Good morning, students. Welcome to B.Sc-I second semester Zoology online class. Today I am going to discuss on the gametogenesis. Gametogenesis. Under gametogenesis, there are two processes: spermatogenesis and oogenesis. Gametogenesis is the process of gamete formation. Gamete formation. Gametes are synthesized. Gametes are formed during this process. There are two types of gametes. Male gamete is called sperm. Their production is known as spermatogenesis. Female gametes are called as ova, and their production is known as oogenesis. So gametogenesis is the process by which diploid precursor cells undergo meiotic division to become haploid gametes. See, gametes are always haploid. Our body cells are diploid. So diploid cells have to be converted into haploid cells. For that, the diploid cells must undergo meiotic cell division. Meiotic cell division. So meiotic division to become haploid gametes. In males, the process is called spermatogenesis and produce spermatozoa. In males, this process is called as spermatogenesis, and what they produce spermatozoa. Whereas in case of females, this process is called oogenesis and produce ova. So the process of gametogenesis occurs in the gonads and involves the following steps. See, the gametogenesis, the process of gametogenesis occurs in the gonads. In testis, spermatogenesis occurs. In ovary, oogenesis takes place. Multiple mitotic divisions and cell growth of precursor germ cells. See, first the germ cells are undergo mitotic division to produce large number of the germ cells, precursor cells. Two meiotic divisions, meiosis 1 and meiosis 2, to produce haploid daughter cells. Diploid cells undergo two meiotic divisions, meiosis 1 and meiosis 2, to produce haploid cells. Then differentiation of the haploid daughter cells to produce functional gametes. Haploid cells undergo modification to become either ova or sperms. Let us discuss the first process, spermatogenesis. As I told you, spermatogenesis takes place only in males. Male gonads responsible for the production of sperms. Male gonads are testes. Spermatogenesis describes the production of spermatozoa in the seminiferous tubules of testis. So testis is made up of seminiferous tubules where spermatozoa are produced. The process begins at puberty when the germline epithelium of the seminiferous tubules divides by mitosis. So in males, this process, formation of spermatogenesis, begins at puberty. Maturation. After maturation only, the production of spermatogenesis begins. These cells, spermatogonia, then undergo a period of cell growth, becoming spermatocytes. Look here, these are spermatogonia. Spermatogonia are diploid. They are diploid. Spermatogonia undergo meiotic cell division. Spermatogonia. So the spermatogonia develop to become primary spermatocyte. Primary spermatocyte, spermatogonia, both are similar. Genetically, both are similar. This is also diploid and this one is also diploid. Only difference is size. This is smaller, this is larger. Primary spermatocyte. This primary spermatocyte undergoes meiotic cell division. This is first meiosis and this one is second meiosis. At the end of first meiosis, two haploid cells are formed. So first meiosis is reductional division. 2n becomes n, haploid. Again, these cells undergo second meiosis to produce 4 haploid cells. So at the end of first meiotic division, 2 secondary spermatocytes are produced. Secondary spermatocytes undergo second meiosis to produce spermatids. These are spermatids, not sperms. Spermatids. These are spherical in shape. After that, they undergo differentiation, cell differentiation to become tadpole-shaped, tadpole-like, sperms. Sperms are produced at the end of the spermatogenesis. So these cells, spermatogonia, undergo a period of cell growth, becoming spermatocytes. The spermatocytes undergo 2 meiotic divisions to form 4 haploid daughter cells, spermatids. The spermatids then undertake a process of differentiation in order to become functional sperm cells. So these are functional sperm cells. Next, oogenesis. So oogenesis describes the production of female gametes within ovaries, and to a lesser extent to the oviduct. So ovaries produce ova. Actually, the oogenesis occurs in ovaries. The process begins during fetal development when a large number of primordial cells are formed by mitosis. So the process of oogenesis in females begins during fetal development, whereas in case of spermatogenesis begins only after puberty in males, but in females during the fetal development oogenesis is started. About 40,000 oogonia are present in each ovary. These cells, oogonia, undergo cell growth until they are large enough to undergo meiosis, becoming primary oocyte. The primary oocytes begin meiosis but are arrested in process 1 when prophase 1 when granulosa cells surround them to form follicles. See, look here, this is oogonium. Oogonium is also diploid. This undergoes a growth phase, larger in size. This is called primary oocyte. Primary oocyte. Actually, oogonia are present in the ovary during the fetal development, fetal growth. So the primary oocyte becomes secondary oocyte only after puberty. Only after puberty. See, all oogonia, most of the oogonia are present in the primary oocyte stage. Primary oocyte stage. But when primary oocyte undergoes meiotic cell division, first meiotic division, prophase 1, at prophase 1, the further growth is stopped. Further growth is stopped till puberty. When the female attains puberty, this stopped oogenesis again begins. So primary becomes secondary oocyte. Secondary oocyte. Okay. So secondary oocyte, and one more cell is released, that is the first polar body. Next, at the end of second meiosis, the oocyte and second polar body is produced. Okay, so this is ovum proper. This is ovum proper. Out of 4 cells, only one functional ovum is formed. Remaining 3 are polar bodies. They degenerate. Whereas in case of spermatogenesis, all 4 cells are functional. All 4 cells that are produced at the end of second meiosis, they are functional and they are sperms. But here in oogenesis, at the end of oogenesis, 4 cells are formed, but out of 4, only one is functional. Only one is larger, remaining 3 are smaller, and they degenerate. But only one, this one is remaining functional. So the primary oocyte remains arrested in prophase 1 until puberty, when a girl begins her menstrual cycle. Each month, hormones FSH will trigger the continued division of some of the primary oocytes. These cells These cells will complete the first meiotic division to form two cells of unequal size. One cell retains the entirety of the cytoplasm to form a secondary oocyte, while the other cells forms a polar body. The polar bodies remains trapped within the follicle until it eventually degenerates. All polar bodies are degenerated. The secondary oocyte begins the second meiotic division, but it is arrested in metaphase 2 again. It is arrested at metaphase 2, means yet the second meiotic cell division is not completed. The secondary oocyte is released from the ovary. This process is called ovulation. So from the ovary, ovum is released at the secondary oocyte stage and enters into the oviduct. Here the oviduct is developed into in mammals. Human beings, the follicular cells surrounding the oocyte form a corona radiata and function to nourish the secondary oocyte. So these follicular cells they form corona radiata and they nourish the ovum present at the center. Okay, corona radiata and function to nourish the secondary oocyte. If the oocyte is fertilized by a sperm, chemical changes will trigger the completion of meiosis II and the formation of another polar body. The first polar body may also undergo a second division to form third polar body. So once meiosis II is complete, the mature egg forms a worm before fusing its nucleus with the sperm nucleus to form a zygote. So this shows the completely formed oocyte, secondary oocyte it is. Secondary oocyte is present in a second metaphase stage. Second metaphase stage is completed when sperm enters the womb. Sperm enters the womb. This is called fertilization. Only after fertilization, second metaphase stage is completed and the second anaphase is completed, second telophase is completed. Later on, zygote is formed, zygote undergoes cleavage. While the process of spermatogenesis and oogenesis follow the same pathway, basic pathway, several key differences exist in between these two. What are the key differences? Number of cells produced. How many gametes are produced at the end of gametogenesis in males and females? In spermatogenesis, the cells divide equally during meiosis to produce 4 functional gametes. In oogenesis, the cells do not divide equally and as a result, only one functional gamete is formed. So, 2-3 polar bodies are degenerated. Then size of the cells produced. Size, whether the cells, at the end of the meiosis are having same size or different size. Size of the cells produced. In spermatogenesis, the cells that are formed following differentiation are all of equal size with equal amount of cytoplasm. In oogenesis, one daughter cell, the worm, retains all of the cytoplasm while the other daughter cells form polar bodies. The polar bodies remain trapped within the surrounding layer of follicle cells until they eventually degenerate. So out of 4 cells, only one cell is larger and 3 cells are smaller. 3 smaller cells are called polar bodies. They are trapped and they are degenerated. Only one cell is functional. Then timing of process. In spermatogenesis, the production of gametes is a continuous process that begins at puberty and continues until death. In males, spermatogenesis begins at puberty, but it continues till death, until the death of a person. Whereas oogenesis, the production of gametes is staggered and finite process. It begins before birth, prenatally, with the formation of a fixed number of primary oocytes, around 40,000 in each ovary. It continues with the onset of puberty according to a monthly menstrual cycle. It ends when hormonal changes prevent the further continuance of the menstrual cycle, menopause. So in females, the production of ovum is not a continuous process till their death. They undergo cessation of menstrual cycle, that is called menopause. Around 45 to 50 years of their age, they stop their menstrual cycle is stopped. That phase is called as menopause. Whereas in case of males, the process of spermatogenesis continues until their death. So summary of the differences between spermatogenesis and oogenesis. So this column comes under spermatogenesis, oogenesis. Process, location. Spermatogenesis occurs entirely in testes. Oogenesis occurs mostly in ovaries. Meiotic division, equal division of cells. Unequal division of cytoplasm occurs in oogenesis. Then germline epithelium is involved in gamete production in spermatogenesis. It is not involved in gamete form production. Germline epithelium is not involved in gamete production. So not all cells, only one cell is formed into gamete. Remaining cells are formed as follicular cells, which surround the centrally present ovum. Gametes, number produced. Here 4 numbers are produced, 4 sperms are produced. Here only one ovum is produced. Then size of the gametes: sperm smaller than spermatocytes, ova larger than oocytes. Timing, duration. Spermatogenesis uninterrupted process. It is uninterrupted, it is continuously occurring. Whereas here in oogenesis, it is arrested stages. It arrested at certain stage, the process of oogenesis is arrested. So onset begins at puberty, begins in fetus. Release, it's a continuous process, and monthly from puberty. Okay, monthly from puberty, menstrual cycle. And this spermatogenesis is lifelong, but reduces. The amount of sperm production is reduced with age. Oogenesis terminates with menopause. So this schematic diagram tells you how the oogenesis takes place. Primary oocyte, then two unequal division occurs. Primary oocyte undergoes first meiotic division to produce two unequal sized cells. One is larger, another is smaller. Larger one is called secondary oocyte, smaller one is called first polar body, and these two cells undergo second meiotic division to produce 4 cells of unequal size. Out of 4, one is larger and remaining 3 are polar bodies. They degenerate, and this will be remaining active. So this is called ovum. This ovum is at secondary oocyte stage, and that would be released from the ovary. That release of ovum from the ovary is called ovulation. So this secondary oocyte is present in second metaphase stage. After the fertilization only the metaphase stage is completed, and this completes its growth. Spermatogenesis, primary spermatocyte, it undergoes first meiotic division to produce two secondary spermatocytes of equal size. Then secondary spermatocytes undergo second meiotic division to produce four spermatids of equal size, right? And they undergo differentiation to become sperm. This is called spermiation. So spermatogenesis. This one is spermatogenesis, and this one is oogenesis. So with this we have completed gametogenesis, spermatogenesis, and oogenesis. Assignment on this topic. So what is gametogenesis? You have to explain it and describe spermatogenesis with a schematic diagram. With schematic diagram, you have to explain it. Schematic diagram. This is schematic diagram of spermatogenesis. You can see here schematic diagram, and this is the schematic diagram of oogenesis, but it is not clear. This you must have studied. You have already studied in PUC. If you refer the PUC book, there it is given nicely. Schematic representation of oogenesis and spermatogenesis. So these are also schematic representation of oogenesis and spermatogenesis, but all steps are not clearly given here. So with this we have completed this chapter. Thank you.